This is the cell cycle. The biggest and outermost circle is the cell membrane. The circle inside of the cell membrane is the nucleus. Inside the nucleus there are strands of DNA arranged into chromosomes. Chromosomes are comprised of chromatids being held together by a centromere. Towards the bottom of the cell are organelles called centrioles. Interphase. Interphase is the longest part of the cell cycle and is divided into three major sections: G1, synthesis, and G2. G1 is responsible for the growth, development, and differentiation of the cell. Each chromosome only has one chromatid, and G1 is 2N, 1X. If the cell passes the all or nothing point known as the restriction point, or G0, then it is obligated to divide. If the cell does not divide, it does not pass the checkpoint. Some examples of this are muscles and nerve cells. After G1 comes synthesis. In synthesis, DNA is copied, for each homologous attached to its copy. 4 sister chromatids are called a tetrad, and synthesis is 2N, 2X. After synthesis is G2. G2 is the preparation for cell division. The G2 checkpoint is where the cell is checked for DNA damage. If there is no damage, the cell goes to M phase. If there is damage, then the cell tries to fix it, and if it can't, apoptosis occurs. Apoptosis is programmed cell death through the method of blebbing. G2 is 2N, 2X. M phase is meiosis. It is the shortest phase of the cell cycle and is where cell division occurs. Meiosis is divided into meiosis 1 and meiosis 2. Meiosis 1 is reduction division, and meiosis 2 is multiplication division. Meiosis 1 goes from 2N, 2X to 1N, 2X. Meiosis 2 goes from 1N, 2X to 1N, 1X. The stages of meiosis 1 are prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. The stages of meiosis 2 are prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. First up is prophase 1. In prophase 1, the nuclear membrane starts to disintegrate. The centrosomes duplicate and migrate to opposite poles. Homologous chromosomes pair up, resulting in 4 tetrads, 4 chromatids, and synapsis occurring. The spindle apparatus also forms during prophase 1. The black dotted line that can be seen in the image coming up is the metaphase plate. Underneath the metaphase plate, arrows can be seen pointing to a centromere with kinetochores. The law of independent assortment states that the orientation of one pair of chromosomes is independent of the orientation of another pair. In metaphase 1, the centrosomes migrate to opposite poles. Chromosomes are lined up in pairs at the metaphase plate. 2 red arrows at the bottom identifying the kinetochore microtubules and the non-kinetochore microtubules. In anaphase 1, the cell elongates and the non-kinetochore microtubules lengthen. The kinetochore microtubules shorten and homologous pairs of chromosomes separate. However, the centromeres do not divide. Next up is telophase 1. In telophase 1, a new nuclear membrane forms. The chromosomes begin to uncoil and the actin microfilament ring begins to form. The cleavage furrow forms, and telophase 1 is 1N, 2X. After telophase 1 is cytokinesis 1. Each nucleus is haploid. The DNA in daughter cells are different than the DNA found in the parent cell. Meiosis 1 is complete, and the cell is divided into 2 daughter cells. It is important to note that cells do not undergo the cell cycle between meiosis 1 and meiosis 2. In prophase 2, the spindle apparatus starts to form, the centrosomes are duplicated, and the nuclear membrane disintegrates. In metaphase 2, the chromosomes line up on the metaphase plate. Their orientation on the metaphase plate is random and independent from the other chromosomes. After this is anaphase 2. In anaphase 2, the cell elongates and centromeres are split, and sister chromatids are pulled apart. The non-kinetochore microtubule is shortened, and the kinetochore microtubules lengthen. After anaphase 2 comes telophase 2. In telophase 2, a new nuclear membrane forms. The chromosomes uncoil, and the rings of actin microfilaments start cytokinesis during late telophase. In cytokinesis 2, The chromosomes uncoil and each nucleus is unique and different from the parent. Uneven division of the cytoplasm, where oogeogenesis goes from 2n2x to 1n2x to 1n1x. And spermatogenesis also does the same thing. To recap, in meiosis 1, there is interphase, and then there is prophase 1. And after that comes metaphase 1. After metaphase 1 is anaphase 1. Then there is telophase 1. And after that, there is cytokinesis 1. Then comes meiosis 2, which is comprised of prophase 2, metaphase 2, anaphase 2, telophase 2, and finally cytokinesis 2.